You don't need to be a full-time biotech analyst to get a sense for the burgeoning RNA therapeutics landscape. The space has been sprouting new company growth at a dizzying pace, particularly on the heels of the COVID pandemic, and it's showing no signs of slowing down. While the development of RNA therapeutics is arguably the brightest spot on the biotech horizon, you might also argue that the public and private research infrastructure required to support the advance of RNA as a therapeutic modality has some catching up to do. I'm Matt Pillar.
And I'm Anna Rose Welch.
And on today's episode of the Business of Biotech, we're going to take a deep dive with a couple of RNA industry notables who are working hard to create a world-class research and development resource for RNA innovators centered around a world-class resource of its own right at Johns Hopkins University's Institute for Nanobiotechnology. If you're into RNA, our guests likely need no introduction. Dr. Jeff Coller is a Bloomberg Distinguished Professor of RNA Biology and Therapeutics at Johns Hopkins University, and he serves on the boards of the Society for RNA Therapeutics and the Alliance for mRNA Medicines. His colleague, Dr. Michelle Kim, is managing director of the new Johns Hopkins RNA Innovation Center, the ambitious initiative the duo are spearheading. A Hopkins molecular biology and biochem Ph.D. herself, Dr. Kim brings more than a decade of research experience earned at Howard Hughes Medical Institute, Circulomics, and PacBio to the party. Jeff and Michelle, welcome to the show.
Thank you. Thanks for having us.
Joining us. We're happy to have you.
Yeah, super happy to have you. Yeah. And
no, we're excited to be here.
Yeah. We had the benefit of a conversation a few weeks ago with Jeff to talk a little bit about what's going on there, and it's a relatively new initiative that we're excited to share with the audience because I think the work that you guys are doing is going to be awfully intriguing to the folks who are interested in RNA, who are Business of Biotech listeners. And I thought,
which should be everyone.
Yeah, I mean,
should be everyone.
Yeah, you might not be an RNA developer, but everybody's RNA curious, right?
Should be.
They
have RNA all throughout them. So
yeah, everyone has RNA.
We are mRNA. Come on.
That's painting with a pretty broad brush.
On a t-shirt, right?
We'll go with it. We'll go with it.
All right.
I thought maybe we'd start with the origin story of the Johns Hopkins RNA Innovation Center. What was the sort of the nucleus of thought, that the kernel of thought that sprouted this initiative?
Yeah, it really started back during the pandemic, and so the origin story really goes way back to like 2012. I first started working on mRNA-based therapeutics back in that time, and the space of mRNA therapeutics was something that most people just weren't paying much attention to. And I had gone to some of these meetings that were very small at the time, only a couple dozen people, and had met some of the big players in that field. In particular, a guy named Mike Houston, who is a CSO of a very small company out of Seattle called PhasRx. And I was talking with them, and Mike and I had some ideas about, how to improve mRNA-based therapeutics. This was more than 10, 15 years ago now. Mm-hmm. And I started working with PhasRx, and sadly, they basically went under. But during the pandemic, Mike gave me a call, and he was now CSO of TriLink. BioTechnologies, which, as we all know, bloomed into this huge organization during the pandemic because they were supplying CleanCap for Comirnaty, or Pfizer's version of the vaccine. And what was clear in some of the conversations that Mike and I had was there's a lot of interest in the RNA therapeutic space, but there's only a few dozen labs on the planet that are even working on mRNA translation, mRNA stability. And so how are you going to develop a new therapeutic modality, which has really become a pillar of medicine now, and innovate that if the academic support network just doesn't exist? And, one of the rate limiting steps, of course, with getting academics interested in this is feasibility and accessibility to materials at a reduced cost, and at a cost point where they can do rapid experiments and try to innovate around the iterative experiments that they would do. And Mike and I started talking about this, and he put me together with the one of the founders of Maravai, which is the parent company of TriLink, a guy named Carl Hall. And Carl and I had a conversation around this where we thought, wouldn't it be great if we could get into academics' hands at a very reduced cost RNA-based molecules that they could then, rapidly screen through, rapidly innovate their technology around, and then do experimentations and continue to evolve from that standpoint. And that's essentially what we did is we created this partnership with TriLink in order to make mRNAs at a clinical grade level, and sorry, at a preclinical grade level, so that our investigators could work on it.
Mhm.
And really, there's sort of two hopes here. One is that since we're working with TriLink and we're using their SOPs, that any molecule that gets developed in a laboratory would be easily moved to a GMP type production facility. It wouldn't be difficult to move that beyond the preclinical stage. But the other side of it, again, is this idea that if we have mRNAs that are at a reduced cost, it allows the investigators to play with it. It allows us to, try different structures, try different modifications, try different, arrangements of open reading frames, whatever it is, in order to improve upon whatever disease it is that they're actually going after. Because, one of the clear ramifications of what's happened in the last 5 years is that mRNA-based approaches work brilliantly for vaccines.
Mhm.
But to get them to that next step, that promise of, cancer neoantigen therapy or gene replacement of monogenic disorders, we really have to innovate in ways to go beyond the biology that is naturally limiting to the mRNA expression space. So in a nutshell, that's really where we're trying to go and why we formed this center. Um, the other aspect of that is, which I think is a very key piece of the RNA Innovation Center, is that we are situated right next to individuals that are world expert in LNP delivery.
Mhm.
Because it doesn't matter how much you innovate in the mRNA space. If you can't get it to the right cells, well, then you're just not going to, be able to do anything. So by having our investigators in the INBT, the Institute for Nanobiotechnology, it really allows us to play these two technologies off of each other and then go after hard to solve problems.
Michelle, how did you end up joining Jeff here at the RNA Innovation Institute? I mean, was it how did this partnership arise?
Quite serendipitously. I
As the
was
best
at
partnerships
PacBio. Yeah,
do. Yeah.
I was at PacBio and I was, well, I should back up a little bit. I was at a startup that rolled out of Hopkins, which was acquired by PacBio. So I really loved the startup environment. And I really love PacBio. It was a great company, but I was searching for something more akin to a startup environment. I reached out to Jeff because I saw that he is active in the startup space, and we got to talking. And he said, actually, I have this other thing that's going on, and you might be interested in that. So,
This other
yeah.
thing.
When was that, Michelle?
Guess that was in the beginning of 24, maybe around
Okay.
January that I reached out to you.
Mhm.
Yeah, I think
Yeah.
it was about January 2024.
Nice.
hmm
So what did it look like to you, Michelle, when Jeff said, "Well, I have this thing." What was the thing at that point? Like, was the thing an idea? Was it were there bricks and mortar behind it? Was there,
Oh, no, sorry. Yeah, no, I think it was pretty clear even at that point that it was going to be the RNA Innovation Center and what the goal was and what the drive was and the vision for how it would grow and what it would become in the future. Right. I think
Mhm.
that was already in place. So
Yeah.
I was very excited because I was trained in grad school as an RNA biologist, and going into my postdoc, I took RNA into a translational research space. And then when I went to the startup, I took a step away. So I always wanted to come back. So this just seemed like an amazing segue to go back to the place I wanted to be. So
Yeah. Very serendipitous.
Yeah.
Mhm.
What was your charge, Michelle? What did you know? What did Jeff say? All right, like we're gonna do this thing, and I want you to be on board. Here's what I'd like you to do.
Do you get to make it up as you go
What are you
along?
doing, Michelle? What are you doing?
What do you do, Michelle?
Jeff's listening with very piqued interest.
I
Now.
know.
What are you doing, Michelle?
Yeah, I'm gonna start taking notes here.
Who did I hire, and what has she been doing?
Well, I like to think of our arrangement, and Jeff, you should correct me if I'm off target. Like Jeff is like the CEO, right, and the CSO, and I'm like the COO, right? And I think our visions are aligned on what we want to do here, and I try to make it happen in the broadest sense of that's how I describe it.
Yeah.
Absolutely.
What would you consider the mission of the center like at this point? You know, you've got the TriLink Association, you've got the infrastructure in place to pull things together, and we'll talk about more specifically about funding here in a minute. But as you formed up, Jeff, how would you sort of crystallize the mission of the center now?
Yeah, I mean, the mission really is to try again in an academic, it's really sort of a quasi academic industrial relationship. And I think that's a very critical piece, especially when you have a technology that really is in its infancy, where you're trying to move it into these new areas. And so its mission is to do just that. It's to take on challenging problems and work with investigators who have expertise in, whether it's cancer biology or some sort of monogenic disorder, to work with them to try and solve a problem. So having, experts in RNA biology, experts in manufacturing, and then experts in delivery, and of course, experts associated with whatever indication, to put those pieces together in a way that is beneficial and can go through experimental reiteration so that we can learn from each other and then gather that information and then hopefully make inroads into this technology. And I really think this sort of model is the only place this is going to really occur, because by the time something gets spun out at the biotech level, they're already set on their way, right? They have whatever product it is that they're thinking about, their indication, they're going down there. There's not much time to go through experimental iterations. And in the academic setting, in the pure academic setting, there's not necessarily the roadmap in how to commercialize and go after therapeutic development. It's really this, it's always been known as this valley of death between academics and industry. And I think what our center does is sort of fill that void in a unique fashion. So our mission is really that, to put into the hands of our investigators the power to be able to try different therapeutic applications and test them, and then hopefully spin those out, either to market them to other partners or to make their own companies associated with Johns Hopkins. So and a long-term aspirational vision that we have is to make our own, to start from ground zero, some sort of therapeutic, and take it all the way to a clinical trial at Johns Hopkins, and have this sort of wholly owned program that we've developed, and hopefully that would take advantage of some of the novel science that we intend to explore.
Yeah, I'd like to add to that too. I mean, Hopkins has a very rich history of taking research from basic science to a therapeutic. And so we would like to be a part of that narrative. And we're hoping to bridge the place where people are at the point of discovery, which, as Jeff said, is more challenging to do if you're in a biotech or a pharma setting. So we can meet people where they're exploring things, exploring ideas, and help them get to the point where they're ready to try something larger in a clinical trial.
Yeah. It begs the question. I'll let you. I know you got one, but it begs the question just for a level set of understanding. You talked about these people, these discoverers, these investigators. Who are these people? Who's invited to come play at the RNA Innovation Center?
All are welcome.
Matt will be there tomorrow at 8 o'clock.
I mean, right now we've started out pretty small, meaning mainly Hopkins researchers, but that's not really small. I mean, we're a pretty big institution. Once we get our sort of the wind beneath our wings, we'll expand from that. But as you know, as Michelle said, all are welcome.
He seemed a little anxious when he said that, didn't he?
Yeah,
Was-
I mean,
He was looking at us in a strange way. Did you see that?
he's recognizing the power of this platform, and he realizes that when this episode drops, the floodgates will likely open.
Well, to be honest with you, the reason I say that is because I have been working on mRNA biology since 1993, for a very long time, and up until 2019, nobody cared about any of that. It was very esoteric and stuck in academia. And I've seen sort of how my life has changed since 2019 and the explosion of interest into the research we do into old things that we had done in the past. And it's a floodgate has opened since 2019, 2020 of people wanting to get into this game. Because when it comes to sort of therapeutic development, as you guys know, you've been doing this for a long time with this podcast, therapeutic development is really very particular, right? You have an indication, somebody discovers a small molecule or, interesting gene therapy, whether that's base editors or CRISPR or something. But in the mRNA space, it's so easy to really think about things that you might want to try, whether that's creating antigens or whether that's, gene therapies as an mRNA or base editors or, CRISPR as an mRNA. It's just really easy for people to imagine things they'd want to do. And so that does open this floodgate of like, well, hey, I could try my favorite thing now. All I need to do is have some tools on how to do it. And that's the piece right now that a lot of investigators are missing, but they're eager to get involved. Even before we officially launched this, the rumor mill in Hopkins was brewing, and we were getting so much interest of people just coming up and asking, could we do this? And my lab, in fact, started making RNAs for people just on the bench because there was too many people asking us to do this. Yeah. So, when I say that we start out small, we have to, because I just think we will get flooded with a lot of requests.
Yeah. Jeff says he's been working on RNA since 1993. I'm like, what? Like you were coloring RNAs with your crayons? Dude, you don't look old enough to have been doing anything since 1993.
It's very kind of you, but it is a fact.
Well, I think, you've talked a little bit about sort of the state of the mRNA space, what it was prior to the pandemic and now where we are today, right? But I think one of the biggest questions I had, when hearing about the center is why now, right? And so I think every good origin story, right, has a why now. And you and Michelle and Jeff and you and I, we met just a couple of weeks ago, right, at the Alliance for mRNA Medicine's first Ascend conference, right? And so we were kind of privy to a lot of the ongoing challenges that the space is having. But I'm curious, how would you answer the why now question? Why do you think that this center is so extremely necessary today, given what's going on in the mRNA space? And what are some of the biggest innovations that really you think need to be made in the space that you think the center will really help pave the way towards? So maybe we can start with Michelle and jump to Jeff.
Sure.
Yeah.
Yeah, I mean, I think this also answers who we're trying to serve. Like we are starting with the Hopkins community because, as Jeff said, there are a lot of researchers here who are interested in working in mRNA or with RNA generally, but they don't necessarily have exposure or molecular biology skill set in their labs. So we want to bridge that divide. I think it's a time where a lot of people can start thinking about this new modality. It might spark some different novel approaches to treating things that previously were not treatable. So I'm so sorry. I just lost track of your question.
No, what's the origin, why now? Why do you think this is an important time for the center and, given this current state of the mRNA space?
Yes, and I think as following up on what Jeff said, because of the pandemic, because of the efficacy and the clearly proven value of using mRNA medicines, a lot of people want to get into this research area. They don't have the skills necessarily, but we do. We can offer them guidance, we can offer them resources, and we can offer them a pathway to taking their ideas from discovery stage to the point where they might be ready to go to a larger scale study.
Mm-hmm.
Yeah, and I'll just, dovetail on that is the when it there's really two mRNA based therapies that are out there.
Mm-hmm.
And they really do represent some of the most widely understood and tested therapeutics that have ever come out. But they're based on sort of one set of rules in terms of optimization of those. And when I mean the manufacturing is one aspect, but then the biology is another aspect. And how many other things are out there that we haven't tried yet, right? I mean, they work really well for vaccines, but mRNA as a drug is a good drug because it's cleared by the body. It's cleared by the body very rapidly. And that's perfect in some applications like a vaccine. It's not great if you're trying to do something like a gene replacement or protein replacement therapy. And then, we're learning other things as we move along. I mean, the pandemic fueled a need to get these vaccines out right away. And so both BioNTech, Pfizer, and Moderna linked, decided to use, M1 pseudouridine as their modification. Well, that might not have been the best choice now in retrospect, because we're getting data coming out in the publication sphere that, those modifications, well, allows for the vaccines to be useful. There could be, unintended read-through events that, we might not want. And there's over 150 unique nucleotide modifications that we know of. Have we explored that entire space? Could we get to a point where we could improve the expression profile of an mRNA and reduce its immunogenicity better than what we already have? And there's real examples where we might need that. So, for example, it's not a case. It's not what we've heard from our colleagues in the government is that the influenza outbreak is not a when, or sorry, if, it's a when, that there will be eventually a large scale influenza outbreak, and we're going to need viable vaccines for that. And in those cases, you often have to mix multiple mRNAs together, but you get to a point where you have too much mRNA per dose. And so that creates an immunogenic response. And so can we explore technologies, modifications, or alterations in the mRNA structure that can reduce the overall amount of mRNA needed to get the same amount of protein product, and then allows for us to go into these more complex therapeutic options? And so I think why now? I mean, that was your question. Why now? Is because we don't have, this technology is powerful. We need it. We're going to need it for rapid responses to pandemics. We're going to need it for other, rare diseases that we might want to go after, or just simply reducing the costs of already existing therapeutics because it's cheap relative to other therapeutics. But the why now is because we don't have the decades of knowledge and how to improve upon the efficacy in vivo, but also the manufacturing. And you could even see from the event you were talking about, the Alliance for mRNA Medicines event, there's so much interest in just the manufacturing alone and how we can reduce the cost and make things better and safer. So it really is an important moment.
And understanding too, I think during that conference, there was a really good quote about how we know. So we've done decades and decades of research into protein structure, right? But there's still very little understanding of RNA structure in and of itself. And
That's
so,
right.
a lot of the work that you guys are really well suited to address, right, is to help us sort of answer the questions that maybe biotechs today are not quite capable of answering yet, or because of different risk tolerance, right? And regulatory tolerance. There can be some limitations there commercially that aren't necessarily faced with academic research. So,
Absolutely.
it begs the question. What Anna Rose was just mentioning begs the question. And Jeff, when you were sort of giving us the big picture earlier in the conversation around what the center intends to do, You hinted at the center being a support mechanism beyond just creating a sandbox for scientific experiments. You hinted at
Hmm.
the translational
Hmm.
capabilities, the relationship with TriLink being sort of foundational to your point, being able to take some of these ideas eventually into the clinic. What does that infrastructure look like at the center right now, and what do you aspire for that? I guess, yeah, infrastructure or functionality to look like in terms of a business support mechanism versus just some lab space and some
Sure. Yeah.
highly decorated scientists who can help you
Yeah.
work on your science experiments.
I mean, the way I see it is so by the relationship with TriLink really gives us a lot of um, how do I want to say this? Well, it's powerful because you're using essentially their SOP to make the mRNA, and it's essentially the same SOP that a GMP mRNA would be made at, but it's not GMP. Um, so what that means is that if the researcher, again, I think I said this before, but the researcher can take it from the preclinical state to the clinical market without having to do too much new optimization. So I think that's an important piece because things can break between preclinical studies and clinical studies, and you don't want that to happen. But I think from sort of the support and business structure it's really this reiterative approach where if you can give an investigator an mRNA at cost, essentially for materials, and then they're testing it and they see whatever it is that they want to see, they might come back to us and then say, hey, we need a little bit more. We need we're getting too much, off-target effects, or the immune reactions are too high, toxins too high, something. So we can, try and play with that to get to a point where they're happy with the outcomes that they're getting, that then might allow them to move on to either new co-startup or licensing it to a pre-existing biotech. And with a product essentially that has been very well vetted in the academic clinical setting, hospital setting here at Hopkins. So I think that provides a lot of value because you don't often see that and certainly, I mean, again, this goes back to this valley of death issue. There's a lot of great ideas that start in laboratories like mine that just wither on the vine, either because they can't leap that valley, or when they do traverse that valley, they die of starvation because there's just not enough, energy to keep them going, to get it into the market space. So I think we can help provide some of that by offering, input and looking at their data, helping to evaluate, and then really seeing what more levers can we pull to get them to the place that they really need to be.
Yeah, that's exactly right. Because if you were to have a company make an mRNA for you, they might charge you a lot of money to get milligrams of one type of mRNA molecule. What we hope to do is be able to charge less because we'd be charging them at cost plus labor. And then we could give them a panel. So instead of getting one type of mRNA molecule, they can get 10, right? And they would all vary in different ways that we think might be beneficial to its expression or stability. And then they can see which one is the best in their assays. So it's a much more powerful approach when they're working with us.
Orthogonal.
And then
And more
eventually
affordable.
yeah, and what we really hope to do is having all this under sort of one clearinghouse is we learn from each of these programs that we assist. So if we have investigators that are doing neoantigen therapy in the pancreas, we take that information back with us and we learn something about what worked there and what didn't work. And then the next person who comes up and wants to, start a program somewhere else, we learn from that. And over time, we're gaining expertise about when do you need to have ultra pure RNA? When do you tolerate a little bit less purity? When, would it be great to have this particular type of RNA modification or this, particular UTR or whatever it might be, or LNP formulation? By gaining that information, keeping it in our records, and then advising, we're educating ourselves, we're educating our own community to help. And so it's creating value to the center itself.
What are the resource? What's the resource picture look like? You talk about the expertise and the research. Are you relying on Johns Hopkins personnel to contribute to assisting with this research and playing a role here, or does the center have its own staff? Like, what do those resources look like?
So in terms of like the financial aspects, this was a joint venture between Hopkins and the Whiting School of Engineering at Johns Hopkins. Between TriLink and the Whiting School of Engineering at Johns Hopkins. So TriLink and Johns Hopkins put in equal amounts of money to launch the center, and it's a 5-year agreement currently. So the employees of the center are employed by Johns Hopkins through that money, but it's as with any academic center, it's never a money-making business. It's basically break even. That's it. And so we charge for our services enough to keep, everything balanced. But we are an academic unit, so we also can apply for grants and take donations. If you want to send us a check, go ahead.
Just gonna slide that little
slide that
info in there.
Send it directly to my home, but no, it's a joke.
So Matt and I are supposed to show up at 8 a.m. tomorrow and also pay to play.
That's right. That's exactly right. That's right. I'll send you my Venmo account.
I'm on it.
But we anticipate that we will have multiple. Like any other academic center, we're going to be applying for grants. We'll be applying for grants with other investigators. And eventually there will be production of IP that will come back to the center. And all of that sort of, hopefully in time becomes self-reinforcing and sustains the operations.
Yeah. Yeah. That's another question I had for you was what claim the center and Johns Hopkins might stake in the potential value of some of these assets that you're helping to create. What does that business model look like, or is it sort of TBD?
I think they're standard. That's all stuff that's controlled by our tech transfer office at Johns Hopkins, which at Hopkins is called the Johns Hopkins Tech Venture, so JHTV. And with anything that comes out of Johns Hopkins that has, that's Hopkins IP, they have the rights over that. And when a new co is started, then there are, the license has to be negotiated with Johns Hopkins. It's a very standard operational procedure for any university and how they spin out companies. And that's really it, is that if we develop things at Johns Hopkins, then if the investigator wants to patent that, then they're welcome to do that. Then it's all done through the terms of Johns Hopkins University.
What about the TriLink influence there? Are there any strings attached to TriLink for these investigators who are coming in and have the potential to take something into a manufacturable state?
I mean, TriLinks, they're very generous in this regard. I mean, they would like the right to look at it, but that's essentially it. Meaning that they have the right to look at it and potentially offer, licensing terms. But our investigators can say no, and so it's very generous.
Yeah, right. They're very supportive.
Yeah.
An important point for any would-be players who are like, what would I be tying myself down to? Yeah.
Well, I think from TriLink's perspective, what they really are hoping for is because the investigators are entering in early in their development and we're using their SOP. So it's the clean cap and the clean script protocol that, they're putting more shots on goal to get molecules into their GMP. We don't really want to start making GMP mRNA at Hopkins. It's a whole different, ball game.
Different ball game, yeah.
Yeah, and since we are using their protocols, we would be sending business to them, and so that's what they get out of it more than anything. So a lot of the de-risking is done what they probably see is that most of the de-risking is done with us through the academic center, and then they get the good stuff
Mm.
Mhm.
that then they can make GMP.
Yeah.
But in terms of IP, it's
Mhm.
there's no tether.
Yeah. So
Yeah.
go ahead, Michelle.
I was just
You're
going to
gonna add
say,
something.
yeah,
Yeah.
TriLink is extremely supportive of this endeavor. And in fact, they had a small team here last week walking us through their SOPs. So, they're
Mhm.
there, they answer many questions, and they're offering their help all the time. So,
Yeah.
yes, the idea is because of this relationship, the transition to larger scale production should be seamless.
Mhm. Mhm. So, Jeff, you'd mentioned that in the past, your lab had been making quite a bit of RNA, right? As things were coming up and questions were being raised and folks
Hmm.
were RNA was getting a lot hotter. I'm curious to kind of get a trajectory here of some of the innovations that your lab has been involved with, right, on the scientific side for RNA, leading up to this point. First of all, where have you been particularly active in mRNA and next gen RNA,
Sure,
R&D?
yeah. Yeah.
Prior to the launch of the center, and then also how you see that supporting some of the work, right, that you're going to be doing moving forward with the center.
Yeah. I mean, my lab has really, for a very long time, worked on stability of
Mm,
mRNAs. That's kind of what our
okay.
bread and butter is. And so, when people have asked us to work with them on therapeutic RNAs, we try to integrate all of our teachings that we've learned on mRNA stability into those molecules. And that's sort of, our knee-jerk reflex is to make
Mm,
sure that we design it the way we think it should be designed, which is not necessarily as intuitive as you might think, as we
mm,
know a lot more about all the various little features that control stability. And it's not as simple as just like going to, some web protocol and hitting like
mm.
optimize my RNA and poof, there it is. There's a lot more that goes into it.
Yeah.
The other side of it is the I'm really fascinated by the RNA modification space
Yeah.
because that is an area we just simply have. Like I said, we're an N of one here, where it's
Mhm.
M1 pseudo U is what everybody throws in for the most part. I mean, there are a few others,
Mhm.
but there's so many other RNA modifications out there yet to be explored
Mhm.
that could really be used for particular applications. And then lastly, the one that you know, Matt was laughing. I said I was been working on this since 1993. It's true. And back then, we were, when somebody would say, because we used to program cells with mRNA back then, we would inject mRNAs into oocytes and stuff and reprogram cells, and people would play with this idea. Could you use it as a therapy? Well, no, because it was delivery, 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 delivery.
Mhm.
Delivery is always the problem. How do
Yeah.
you get the transcript to the cell that you care about? And back then, nobody was really even thinking about the LNP space as a way
Mm.
to um, not only deliver an RNA, but also to optimize delivery to specific cell types as well as affect durability and efficacy. So what's really exciting to me is to have the center right next to these researchers that are world-class LNP experts and start exploring that space together,
Mhm.
because it is true that we're not going to innovate unless we can do that.
Yeah.
So that's another place where we've had some success, on this sort of individual basis of, here's an RNA, here's an application. Let's talk to our colleagues. Do they have an
Mhm.
LNP that might work? And all of this will have to be sort of combined as things change. For example, if you start changing modifications on an mRNA, that's going to change its chemical property.
Mhm.
As so how that gets packaged into the LNP is
That's
going to be different.
huge question.
And
Yeah.
yeah, so how do you have to optimize in that space. You
Mhm.
can't just say, hey, I just discovered this novel way of modifying an mRNA that really works great, but
Mhm.
you try to package it into a generic LNP and it doesn't do anything.
Yeah.
So having those pieces together is really important.
Mhm. An integrated
Yeah,
approach for drug product
exactly.
development. Yeah. Michelle, did you have anything to add? Or you have a background in the RNA world, right? So I mean, are there innovations that you are most excited to see the center tackle in the upcoming year or two as you set out to be COO, right? To operationalize all of these all of the goals you guys have. Are you most excited about anything that you guys are going to be working on in particular? Any innovations you want to see made?
Well, I mean, we'll start with mRNAs, and Jeff's lab has made a lot of very valuable discoveries in how to increase the stability. And as Jeff has often said, if stability is increased, then you're going to have expression. So this is going to be really important. There were a lot of things that I saw at the meeting in Boston last week. Was that last week? 2 weeks ago?
Feels like it. It was 2 weeks. Yeah.
There are a lot of people. I know it really does. A lot of people doing work on self-amplifying RNAs. So that's an intriguing area that we don't have experience in hands-on yet, but a lot of people are asking about that.
Yeah.
And that also is going to have issues with packaging because those tend to be much larger molecules. So I'm really eager to see what happens in that space. And just how mRNAs are used in areas where they haven't been used before, such as like neoantigen therapy. Sorry, I'm getting a cold.
Oh no,
you jinxed me.
I wonder where that came from.
Yeah. Or for delivering enzymes and base editing and CRISPR editing. So I think a lot of people are trying to switch to that. So I think that's a really important area. We're probably going to see a lot of people asking us about that. Yeah. But yeah, I think self-amplifying RNA is an interesting space. I know Jeff and I have talked about that before. It has its pros and cons, but it's nice because you can deliver less and potentially get more expression. But
and potentially unmodified. There's a lot of hope that you can get the RNA to just simply be unmodified and reduce the overall, because again, this whole idea of reducing the effective dose is very important. If you're stuck at like 100 micrograms of RNA, which is what Moderna's was at, it really kind of puts you at that's a lot of RNA.
Yeah.
But if you can get it, and what I've heard from some of the self-amplifying folks is that they're down to like 0.25 micrograms. So it's a pretty significant, difference. And and that puts you into a new realm of where you can start mixing and matching different strands for complex, processes, whether that's like some sort of gene editing or if it is a pan viral vaccine that you want to generate, so you can have 5 or 6 different transcripts in there. To try and get the best response that you can possibly get. So those are all things that we really need to explore.
Yeah, and these explorations, I mean, I'm reading between the lines as you talk about them, and their translation to the business aspect, right? Like, this isn't just a science experiment. This isn't just doing cool stuff. It's like, these are things that we need to do in order to move the business forward. And I'm curious about that, because I don't think it can be overstated that this isn't just an academic exercise, and that you two both bring to the table a bunch of industry and vendor supplier experience. You both worked in the biotech space. You've got, like I said, supply experience. So I'm just curious about what kind of mind share that industry experience you're bringing to bear as you form the center up and create the vision and make it what you intend it to be. How have those experiences that you've had in the industry, the business of biotech, informed the way that you're setting up to help other companies build and grow?
You want to start there, Michelle?
Sure. I think Jeff handles that at the macro scale, and I handle that at the micro scale, which is to say, Jeff established the center. There are a lot of players involved, not just TriLink and Hopkins, but all the individual players within these 2 different organizations. So bringing the center in existence was that was no small feat. And then here, getting operations going at the micro scale, I think because of my experience before in a small startup and then in a larger company, it was an eye opener to me coming from grad school and a postdoc. Excuse me, where you're in a very academic mindset. It's very different than when you're in a company. Like when you are trying to sell something or help people, you have to be very customer oriented. You really need to know how to keep stakeholder engagement. You need to know never to say no or never to say it with that word. You need to understand you're trying to understand what do people need, and then how can you deliver to them, and how can you describe to them what you can and can't deliver. So I think those are the skill sets that I bring from my previous experience. And I mean, I hope it pays off.
Yeah. So
yeah,
far,
I mean,
so good, right?
yeah, I can just to add on that. I mean, I think so. I started my first biotech company back in 2018, 2017, and it's Tevard Biosciences, and I started that company with Harvey Lodish, and he has really been kind of he's a sort of a icon of biotech development, and I learned a lot from him. But one thing you do learn in talking to investors and talking to other players in the space, leaders in the space, is, and it goes to why this center really is important, is that you have to hit the ground running when you start a new startup. You have a very limited time. And so you have to identify a niche that you can occupy, that you bring value to. And that's really where I think we can help in this arena, is if you have investigators that are coming to us and they want to do this, that, or the other thing, it's fine if you're getting a publication, great. But if you actually want to spin that out to make a company, well, then what is the real value of that product? What does it bring to the table? And we can help with that narrative by saying, well, we understand the mRNA space, and we know that, that you have lots of competition over here, but you might have a singularity here that could be explored and provide value that investors would be very interested in seeing. So, we don't work alone in that regard. I mean, we have the tremendous support of, Johns Hopkins Technical Ventures, and as well as our own, contacts in the field. So I anticipate that the center will provide, we do more than just science at this center. You'd mentioned we're part of this Alliance for mRNA Medicines, and that is another aspect, because it allows us to really connect people with other industrial leaders. And we have over 60 to 70 members of that alliance now that we're part of. And that network is very valuable and will be valuable to our clientele as we move forward.
Yeah. I was, you'd mentioned something too about how you can kind of help, your knowledge of the mRNA space can help folks connect their product or their research, right, to some of the overarching needs of that space. And there's a lot of best practices, I think, in terms of, how you can launch a business, and best leadership practices that are really modality agnostic. But I'm curious, with your background, both of your backgrounds being in this RNA world and having seen the RNA world pre-COVID, when no one really gave it the respect that it was due, right? Are there any RNA specific insights that you think are really important for folks to keep in mind as they are launching an RNA business? I like to kind of dig into some of the individual nuances, right, that come from modality specific learnings and running a business.
Yeah.
So,
I mean, I think what we can see from what's happened over the last 5 years, the use of mRNA in vaccines is never going to go away. That's never going to go away.
Yeah.
That's something that, that genie's out of the bottle, and there's a lot of issues around vaccines and political things, blah, 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 but it's not going to go away. It's just too easy, works too well, and not that it works too well, it works great. But I think where we're really seeing incredible promise is in neoantigen therapy
Yeah.
or mRNA. Again, the idea that you can design a personalized therapy for an individual with like pancreatic
Mhm.
cancer and have that, the successes that we're seeing in the last 2 years in pancreatic cancer with neoantigen approaches is really tremendous.
Mhm.
That, this was a disease where it's really a death sentence, but was leading at least to half the people in that study were still alive several years later.
Mhm.
Um, and so that's going to continue.
Yeah.
And there's a very clear path to using mRNA in the neoantigen approaches. It's not going to work. The mRNA is another modality in
Yeah.
our therapeutic arsenal. It's not going to work for everything,
Mhm.
but it is going to work for these other applications. And I don't think this is a crazy vision. I think you're going to see in the next 5 to 10 years sort of point of care therapeutic
Mhm.
development, where you have major medical centers like Johns Hopkins or Sloan Kettering or Houston Methodist creating their own programs to make personalized medicines on site,
Mm.
where you get diagnosed with some rare disease or rare cancer. You go there and they help design a therapy in the house, and you get that therapy. Now, how that all gets paid for, that's a different issue. But the technology is such that we can do it as point of care,
Mhm.
and that's going to expand. That will expand beyond the medical community, also into like agriculture,
Yeah.
and starting to treat um, our food supply before some of these pathogens emerge into the human population. mRNA is a very low cost, viable approach to start, you know, using on some of our livestock and animal products. So,
Mhm.
Yeah,
um,
wouldn't that be amazing? Sorry, if we could decrease the use of antibiotics
Yeah.
and
yeah,
yeah, animal farming, and then maybe reduce the use of pesticides.
Mhm.
yeah, exactly.
Yeah.
It's something we often don't talk about a lot in biotech is where, you have certain approaches like mRNA that can cross just from, you don't necessarily have to apply it just to humans. That
Yeah.
technology is just as viable in other animals. So,
Mhm.
and because it's low cost, that reduces that barrier. So,
Yeah.
Yeah. Jeff, you talked about the valley of death earlier, and you talked about the likelihood of death, even if you make it across the valley of death. You're a very uplifting man, Jeff. You're very inspiring. I'm just kidding.
There's a movie coming soon. I feel like.
I'm curious though, and I mean, you talked about some of the causes of that mortality, but I'm curious if you've seen a lot of this, like you've seen a lot
yeah,
of academic exercises that showed glimpses of commercial promise and opportunity, and you've seen them go north and go south. If
yeah,
if and I'm going to ask you a hard question to answer right now, and I
sure.
apologize for it in advance. If you're going to put your next paycheck on a bet, right? Like,
We
hey,
really are going hard here.
yeah,
There you
like
go.
there's there there's
Michelle, you want in on this too? You gonna risk not feeding your family for a week? Do this?
you're raising the bar way too high for this question. It's not that good. But
Your
it is
delivery.
challenging to answer. If you had to, let's say you got 6 ideas in the lab right now. Each of them have their own own glimpse of commercial promise down the road. But you got to put your money on what's going to go wrong, like because you've seen it go wrong before. Like what's
Yeah.
that knowledge gap or that that missing piece that so often dooms these projects early.
Yeah, well, the biggest thing that dooms almost any biotech is, efficacy versus tox, right?
Mm.
Like how much benefit do you get for the risk, and is that tolerable? Um, and we see this all the time where, there's gene therapies that are going to do great things, but then we get them into the clinic and there's very little success. We've as everybody's said, and you've probably heard a million times in this podcast, is that it's really easy to cure a mouse. We've eliminated cancer and most genetic diseases in mice 1,000-fold. But applying academic research to the human body is much more difficult. And so getting good efficacy and low tox is what is critical. Without that, you don't get out the door. And so if I was betting, in the place where I think a lot of people have made these bets historically is always in the cancer space, because the risk to benefit ratio is so different versus other places. Prophylactic vaccines, you've got a huge, risk benefit analysis that has to be done. You cannot cause harm there at all. And it has to be efficacious, and there's a lot of political aspects about that too. But in the cancer space,
oh really? I haven't heard.
Well,
any
the whole
influence on the vaccine space.
we
Not
could
at
go
all.
down a whole different podcast for that one if you wanted to. But
Coming soon.
coming soon to a podcast near you. But cancer, if is in betting, where the next sort of wildly successful mRNA therapy is going to be, is in these really hard to get to cancers, these recalcitrant cancers like pancreatic cancer, glioblastoma, where we can try, we can take a lot more risk there because the benefit is so high. And that allows for innovation that then can eventually allow us to do things like monogenetic gene replacement, and which has its challenges. The challenge there is the durability of the mRNAs. The mRNA, the reason why I study mRNA is because it goes away. That is my job. I've studied mRNA stability for decades, and it goes away, and it goes away fairly quickly. And so from a protein replacement strategy, unless we improve the durability, or we improve or if it's an indication that allows for repeat dosing, then we're sort of limited there. So from a betting standpoint, if somebody had a really good cancer neoantigen treatment, that's where I would be betting.
All right.
Yeah.
You heard it here first. Anna Rose,
but these are just I provide no market advice.
Remember, there's a paycheck riding on this. So if anyone's listening who pays Jeff's bills over here, we've
Jeff, we'll put a disclosure in the show notes.
Okay, yeah.
Protect you from any advice.
Any legal responsibilities.
yeah, right.
We're gonna need
I
to
know.
land this
I
airplane.
know.
Rose, so
We'd
I'm
love
gonna-
to keep the planes flying. Here,
I've got a couple more, but I don't want to give you short shrift.
No, well, so I guess it's more of an overarching question, just sort of, we've talked about why academia is a great place to do some of the R&D that we really need today, right? And so I'm curious, just as we're looking forward into the future, and just in your experience so far with the center, have you seen an increasing amount of interest from industry in partnering more closely with academia over the past year, 2 years? And do you see that, now that the center's fully fledged and formed and exists, do you see that interest continuing to increase?
Yeah, I think so. Yeah, absolutely.
100%.
Go ahead, Michelle.
Yeah.
Yeah, I mean, I think that's definitely evidenced here with our partnership with TriLink. But when I was in PacBio, I mean, they and other companies that I had spoken with, like everybody is understanding or realizing the real value of academic centers for discovery, right? Because, as we've talked about earlier, it's an inhibited prospect if you're in a biotech company or pharma company, when you have timelines and deadlines and programs. So I think companies are going to continue to grow their interest in trying to partner with academic institutions to help foster these early ideas, and with the idea that it's going to come back to them so they can work on the other end of the research pipeline.
Yeah,
for sure.
Yeah, and this modality really is sort of unique among all other modalities out there, because it's not like if you're doing a small molecule, how are you going to reiterate that and learn. Where here, it's a basic building block. It has to do one thing, which is to make a protein.
Mhm.
And so there's a rule book for that.
Mhm.
And, but we can, it is the messenger of the genetic code, and we can get it to communicate any information we want. And so that's where, why industry is so fascinated by this technology and why they're approaching places like us in order to say, well, what can we do together? And yeah, I only see that interest continuing to go up and up and up in the next few years. This is a technology that's simply not going to go away. It's not.
I like to hear that.
Yeah. Well,
At the helm of a publication called Advancing RNA, we certainly hope it goes nowhere.
kind of your wheelhouse.
Yeah.
What's the next big agenda item for the RNA Innovation Center in terms of its growth and development and go forward plan?
Well, as of Thanksgiving, we are fully operational. So, because we were in the building stage until right now. So we have a number of projects that are on deck. We're gonna be able to dive into them now. So that is the first step in activity, I guess. Yeah.
And as we grow these smaller individual faculty-based projects, we want to expand out from there. And we have some conversations going on about how to leverage this technology in unique ways. Like we're talking with the Applied Physics Lab here at Johns Hopkins on maybe there are things that we can do outside of medicine, more in sort of the technical space, and continue to work with our partners in the federal government to push this technology in a unique fashion. So we're really interested in partnering with groups that want to take on large problems, and because I think that's where we're particularly good, or will become very good at, is not just meeting the needs of the individual, but also of the bigger industry as a whole. So
yeah, where should those groups, those potential partners and researchers who are interested in learning more about what you guys are doing, where can they learn more? Where can they be in touch with you?
They can either contact me or Michelle directly, or we have a website.
We have a website.
Yeah.
And a LinkedIn page.
All right.
Many avenues.
Maybe some T-shirts in the future.
Yeah, we're gonna get T-shirts. We'll
I'll help you. I'll help you come up with slogans. You're welcome in advance.
Don't forget the link to the Venmo. Okay.
Yeah. Just personal Venmo at the bottom of each T-shirt.
I think I
Chief Marketing Officer.
Send me your Venmo. We'll put that in the show notes too.
That's right.
Michelle, you want to get in on this too?
I guarantee the
100%.
Yeah, I guarantee the inbound interest in the offering is going to far exceed the inbound Venmo, but we'll put it out there.
That's fine.
Well, guys, this is I feel like we've just scratched the surface. Michelle, to your point, you're fully operational. There's some projects in the works. We're going to have to revisit.
We have to have you back, man.
We're gonna have to revisit down the road and learn about some of that project work and stay up to date with the progress of the RNA Innovation Center, and any number of these things. The vaccine conversation. I'd welcome that.
Yeah.
Vaccine politics.
This is
There's a lot. Yeah.
Today's reputation is it's a hot topic.
I mean,
I'm here for it.
I was
ready.
sitting here dreaming up an episode dedicated to the application of RNA technologies in veterinary science as a means of reestablishing public trust.
Yeah.
I mean, there's a lot to talk about there, right? Like
there is a lot to talk about, and there are people that are trying to help in this space. That's what, in part, the mission of the Alliance for mRNA Medicines is about. To really try to, change the public narrative. We have a lot of challenges ahead of us, and it's important that the public realizes that these technologies are not there as they're not bad technologies that are meant to just make people money. They are technologies that actually are saving people's lives, and they're going to become very important in the next few years. And and it's a technology that really started with, Operation Warp Speed during the Trump administration, and it was launched during that time, and we have to embrace it, and we really have to rally around it as a technology.
It's here to stay.
It's here
in
to
us.
stay. Yeah, it's in us and it's here to stay. So
Yeah. Well, congratulations on the progress you've made with the Innovation Center to date, and the role that you're playing in that messaging and that work. And like I said, we're definitely going to have you back on the show. I've heard that you guys and Anna Rose hang out on the road quite a bit. So
this is the next time we do this. There will be Manhattans and perfect Manhattans and other forms of bourbon involved, because that's when the conversation gets
let's do it in Manhattan.
really lit.
Yeah.
Ooh,
I'm there. I'm
go.
there.
Traveling.
I wasn't going to bring it up. You
Okay.
just did.
Yep.
But
Here we
yeah, she'll let me know, I'm sure, when you guys are ready to come back on and share an update. But in the meantime, thank you both so much for coming
thank
on
you.
and sharing the story.
Thanks for having us.
Thank you. It was fun. Thanks a lot.
So that's Doctors Jeff Coller and Michelle Kim. I'm Matt Pillar.
And I'm Anna Rose Welch.
And you just listened to the Business of Biotech. If you're working in the RNA therapeutic space, or simply, as I said earlier, RNA curious,
Which you should be.
Anna Rose and her work at Advancing RNA are must follows. So bookmark advancingrna.com, or you can subscribe to Anna Rose's newsletter and connect with her on LinkedIn. And if you're loving the Business of Biotech, make sure you subscribe anywhere you get your podcasts, and go to the videocast page under the Listen and Watch tab at bioprocessonline.com. Where you can not just listen to Anna Rose and I banter, you can watch us.
I know. It's
Pretty
really,
cool.
it's very exciting.
We drop a new episode every Monday morning, so we'll see you then, and thanks for listening.